Myra Fernandes in psychology. Title is Using Neuroimaging to Understand Cognitive Brain Function. Myra, welcome.
Okay, so thank you very much for inviting the folks over in psychology over to tell you a little bit about what we do over at our end of campus. What we look at primarily is how to use neuroimaging to help us understand higher cognitive brain function. What I mean by higher cognitive brain function I'm going to focus in on three things, and that is how do we represent different classes of information in the brain. The second thing is language processing, and how can we use imaging to help us localize language in the brain. And finally, memory. How can we use neuroimaging to help us understand the brain regions that help us perform different memory tasks, and how do those break down when people get older, and in particular in senior citizens. The first cognitive function, as promised, is visual processing. And one of the questions that we ask is, do we have unique brain regions devoted to the visual representation of words, digits, and other symbols? This is a question that is primarily of interest to us for understanding how it is that we come to have specialized brain regions devoted to the processing of different classes of stimuli. What we do in these sorts of paradigms is we have people go into a scanner, a 1.5 Tesla scanner. This is work that was actually done at the Rotman Research Institute through collaboration with Sunnybrook Hospital. And we put people in there and we present them with different classes of information. What we wanted to find out was whether there is a brain region devoted to the visual representation of words, which we have become experts at recognizing, as opposed to symbols, digits, Hebrew and non-speaking Hebrew people, and control visual dot patterns. One of the ways that we do this is we overlay traditional MRI images. We overlay functional images on top of traditional MRI images to yield what's called differences in brain signal activation, or the BOLD function, as Rich Daines outlined to you in an earlier presentation. And what we want to know is this area on the left side of the brain in red is known in the literature as a visual word form area. It's been purported to be selectively active for words, which seems a little odd to us given that the processing of words and language is a cognitive function that develops as we age. Would we really have a brain region that would be devoted solely to that sort of function? We thought not, which is why we included symbols, to look at whether the brain is specialized instead at extracting information from known abstract stimuli. So looking back at the images here, the symbols are not words, but they are abstract stimuli with which we can extract meaning. And what we see in the top is the left fusiform area. This is the coordinates of the visual word form area. And shown in blue and green, what you can see there is that activation for words and symbols follows a similar pattern of activation that is distinct from the activation that you see from digit control symbols or Hebrew language. Well, this is what I call a hotspot approach. One of the things that we're hoping to develop a little bit more in my lab in particular is how to use imaging data to give us that much more information. So we don't just have one brain area participating in a particular cognitive function. There's presumably lots of different areas that are participating. And how can we use that information to tell us more about brain functioning and cognitive functioning? So in this sort of analysis, the partial least squares analysis, what we're looking at is the covariance between brain regions in different areas. So that even though a particular hotspot might be lit up when you view a particular type of a stimulus, what you can perhaps come up with in this sort of analysis is a unique network that is positively correlated with one condition, viewing of words in this case, versus another, which was the viewing of symbols. So we can see here that we can identify a locus, a whole bunch of areas that together co-vary in the word condition as opposed to other conditions. So it gives us a little bit more than just looking at the traditional approaches. And this is something that would be great if there are other people interested or have techniques that can help us to look at these covariance matrices. It would help us to develop better images and overlays. The other area that I'm going to briefly talk about is language, and this was referred to in an earlier presentation. This is a technique that's primarily used in hospitals. You don't just volunteer for this sort of thing. Where what you want to do, and this is used particularly in kids with epilepsy, pre-surgical evaluation to find out which hemisphere language is represented in. And here you inject sodium amytal, which is an anesthetic, into one half of the brain, and this just means that that half of the brain is asleep. And what you do is you look at the remaining language function of the other half of the brain to get information about which side is primarily necessary for language representation. As you can imagine, this is very invasive, and other techniques include behavioral tests, such as we use in psychology, where we look at what's called a dichotic listening test and how well we can predict language function based on the what we call eerie symmetry scores from this sort of test. And in that sort of test, we can deliver it to people who aren't in hospitals, who are in schools, or for research purposes, we want to find out what hemisphere language is represented in. But in this sort of test, what we need to know is what is the concordance between some of this imaging, between some of the WADA tests and this dichotic listening test, and whether we can use neuroimaging to help localize language and show that our behavioral tests are actually indicating correctly where language is located. Here's a patient, a child actually, LY, classified with unilateral language representation on our behavioral tests. They show unilateral activation in the scanner so that we can have some confidence that our behavioral tests are actually giving us concordant information. And similarly, here's another patient who was classified with bilateral language representation on the behavioral test and bilateral language representation, again, on the imaging test. So we can develop less invasive techniques. And finally, we can use memory. We can study memory by looking at brain performance scores. This is simply looking at young adults and senior citizens to find out whether there are any differences in memory performance. And you can see that they can perform similarly on a test. But if you look at brain imaging data, what you can find out is that areas shown in red are the brain areas that a senior citizen would recruit for a given memory test, and areas in blue when you're a young adult. So even though they show similar performance, you can have wildly different brain activation patterns. And it would be interesting to know if the recruitment of these additional areas is actually compensatory to memory or in fact redundant, whether they're spinning their wheels. And the last thing that we're doing is try to overlay many people's data onto one brain to look for commonalities in findings across studies so that we'll have more confidence in some of the results that we're getting. Thank you very much.
The things you are doing are so relevant to the discussions that we had with Vlad Ajinsky a few weeks ago. We just have to talk with you and talk with you about what that was a very interesting talk, which is looking at the relationships among stroke and Alzheimer's and ALS and so on, and with the idea, fundamental idea of the ability to prevent stroke in people. But they found very many similarities, including differences in cognitive function, memory, and so on. So objective things like this could be very, very, very valuable. Comments or questions? Eric, and then...
Last slide that you showed, is that an average brain?
What this is a 3D rendering of a brain. It's a blow-up brain. It's a standardized brain upon which we have overlaid the coordinates of activation from about 11 different studies to look at the variability across different studies on what is the same actual task. So you can see there's a lot of variability. No, that's usually what we do, actually, but because this is a meta-analysis, that is not what was done. But usually what you do in these sorts of studies is you collect data from about 10 or 12 people in the scanner, and you should be presenting your data on the averaged neuroanatomical MRI. But what some people do in studies is they pick their best brain and overlay the images. But that's not quite so honest.
Right. I was just wondering, because, someone can give me a picture of an average cell. If I could give you 50 pictures of a cell and you can generate an average image, that would be useful for me to tell averages.
I have some average brains, if you're interested in seeing what they look like. They're a little fuzzy.
I would go for a standard one, average one, even anything. Other picture.
Can I ask a question? I think that, this intelligence, memory, language, they are very complex things. And I think that probably the more important thing is the network. This is about chemical reaction from layer to layer. And here you try to correlate, this sort of memory intelligence with a certain particular physical location of the brain. Do you think that would, correlate well? Because I just saw so much in the thing.
Yeah, this is a very good question, and that's actually why some of the earlier work that I presented here on what's called PLS analysis is try to overcome that particular problem where I'm aware, and most of us in psychology are aware, that there's a whole bunch of brain areas that are probably contributing to function, And so by looking at comparing different tasks, for example, looking at different types of visual information, how those conditions lead to different groups of areas acting together is some of the ways in which we'd like to proceed in the future. And this is the sort of thing that could benefit from collaboration from people here, because right now this is done with collaborators in Toronto at the Rotman Research Institute and Toronto Western Hospital.
There's also an issue in what you're asking about in terms of normally taking a group to analyse what's happening in a given task, because you can get a very nice clean map of the usual suspects of the areas of the brain that are important for completing a whole range of different tasks. If you then look at individual subject maps, not everybody shows those usual suspects. And a question that being asked by some, but probably not enough people doing this kind of research is, why is that the case? And what can I learn from looking at the individuals and how they then relate to that kind of group average map?
And that's actually the direction that most imaging studies go, is that you simply don't look at one or two people. You look at the averaged activation and try to make conclusions from that. And you also trace it back to lesion work with patients who we know brain damage to this particular area leads to this type of cognitive deficit. We start looking around for brain activity in that particular area that we know from lesion studies is a candidate.
All right, thank you very much. And so we also have brain activity for listeners.